Okay, it's another fine day that I want us to look at bacterial infection, which is part 2 of the series of this particular area, which is the bacterial infection. Bacterial infection is one of the most common condition that is found in the outpatient clinics. So majority of cases diagnosed in the outpatient departments is basically the bacterial infections. So we just want to look at it and see how does it present and other related issues revolving around bacterial infections. So these are some of the questions that at the end of the presentation we ought to be able to answer if time allows. And the majority of these questions are usually asked by our patients every time they visit our hospitals. Whenever they are told they have a bacterial infection, they always ask which bacteria. They also ask which type of bacterial infection and how did it get into my body and how did it cause this infection, and so on and so forth. So today, as we proceed in this particular presentation, majority of these questions will be answered. So how do we define bacteria? Bacteria is a microscopic single-celled organism. It exists in their millions in the environment, both inside and outside the human body. While many are harmless or beneficial, some are pathogenic and they cause diseases. So in this definition of bacteria, one of the key words is microscopic, meaning bacteria are very, very small organisms which you cannot see by naked eye. Therefore, you have to use a microscope for you to be able to see. Another key word in this definition of a bacteria is the issue to do with single cell. Single cell. This means bacteria are organisms, but they don't have many cells. So it exists as a unitary cell, just one cell. So it's an organism that exists in one entity, which is a cell. Unlike other organisms like human beings, which have multiple cells. So bacteria is microscopic and is single-celled. And they exist in millions, ubiquitous. They are found everywhere, everywhere, everywhere in the environment. So bacteria are found everywhere in the environment. Everywhere means these bacteria can be found in water. So it can be found in water. They can be found in the air. These bacteria can be found in the air. They can be found inside our body. Can be found inside our bodies. They can be found on surfaces. So the word is everywhere. Everywhere. The mode of entry in the body may differ. Sometimes patients ask, how did this bacteria get into my body? And you see, we are saying these things are found everywhere. Therefore, if they are found everywhere, then there is a possibility that either you got it through air, that's inhalation. You can also get a bacteria inside your body through ingestion. This means you've taken it through water or even through food. So here, it can be through water or it can be through food. Bacteria can also be found on the surfaces of human beings, on the hands, contaminated hands, and so on and so forth. So the issue of where did this bacteria come from is answered here. Bacteria are everywhere in the environment. The only issue is that you cannot see them using your naked eyes. So they are microscopic. So that is what a bacteria definition entails. So what is a bacterial infection? After knowing what a bacteria is, the next thing is to know what is a bacterial infection. And we are defining bacterial infection as an illness which is caused by the invasion, rapid multiplication, and spread of harmful bacteria within the body. They often cause damage in two ways. One, rapid multiplication, and two, through the release of toxins. So this is how bacteria cause infection. One thing you need to remember is that not all bacteria are pathogenic. Some are helpful in the body. We call them normal floras. You can call them normal floras. Sometimes we call them commensals. These normal flora sometimes they also form part of some of the probiotics. We call them probiotics that are given in patients who have developed severe diarrhea, which is acute, and they have lost most of their normal flora. Therefore, they are given probiotics which contain normal flora to replace what has been lost through diarrhea. Remember, some of these normal floras are important in human beings. Some of them in the GIT participate in production of some vitamins. Some of these normal floras. So, not all bacteria are pathogenic. Some of them are helpful, and therefore that is a very important thing to know. Pathogenic or harmful bacteria are always managed using antibiotics. Antibiotics, these are just drugs that have been formulated and they have the ability to kill bacteria. We shall have to see in the subsequent presentations how antibiotics work. We shall look at them in terms of their definition. We shall know what antibiotics are, and we shall know their classification. We shall know classifications of antibiotics. We shall know their mode of action. We shall know what we call the pharmacodynamics. We shall know the pharmacodynamics of antibiotics. We shall also know something about pharmacokinetics. We shall also, when we will be looking at antibiotics, we shall also define terms like potency. We shall also define terms like efficacy. We shall know also things to do with what we call first pass metabolism. And so on and so forth. So these ones are spared. We shall look at them when we'll be looking at antibiotics. And that is to say, that is a topic for another day. So pathogenic or harmful bacteria are normally treated using antibiotics. In the definition of bacteria, some other aspects to remember is that bacteria are prokaryotic. Prokaryotic means they lack a nuclear membrane. So their nucleus, as you can see, is just found in the cytoplasm. That's why they are referred to as prokaryotics. Those whose nucleus has a nuclear membrane, we call them eukaryotic. So mark the difference. Prokaryotic, they lack a nuclear membrane, while the eukaryotic, they have their nucleus enclosed in a membrane. Also said they are unicellular, one cell. They are also microscopic. You can't see them with the naked eyes. Their structure possesses cell walls. And they also have something called peptidoglycan. We shall see. Their habitat is ubiquitous. Meaning these bacteria are found everywhere in the environment. Everywhere. They are found everywhere in the environment. It can be in the soil, it can be in water, hot springs, and organisms. And then when it comes to metabolism, it varies. It can be, you have those who do their metabolism through autotrophic, and you have those that carry out metabolism through heterotrophic. Autotrophic simply means they can synthesize their own food. Synthesize. So those bacteria which can synthesize their own food through metabolism, that is what we refer to as autotrophic. While heterotrophic, they eat food that is already manufactured, that is already produced, or that is available elsewhere. So that is the difference. So bacteria looks like this. This is a diagram for bacteria, just to show how bacteria look like. You can see these projections you see here. These so many projections are what we refer to as the fimbriae. Function of this fimbriae is for attachment on the host cells. So when the bacteria gets into your body, after you have consumed it through water or food, it will attach onto your cells using these tiny structures you see here, what we are referring to as the fimbriae. Then you can see the red projections here are just the flagella. This one helps during movement. You see it looks like a tail. So it helps bacteria to swim like a fish in water. So the flagella helps the bacteria when it comes to movement and navigating through extracellular matrix of human tissues. This is another diagram of a bacteria. Still we have a flagella there. These other structures you see inside here are just organelles, the normal organelles. Like the DNA substance here, you can see it. It's not enclosed in a nuclear membrane. It has a plasma membrane. It has a capsule. It has a cell wall. Remember, human beings lack cell walls on their cells, but bacteria have cell walls. So that's another difference, which is usually considered when it comes to manufacturing of some antibiotics. We shall see this when we will be looking at antibiotics. There's a concept of selective toxicity. Antibiotics, remember when this bacteria gets into your body, it is inside in your extracellular matrix. Extracellular matrix. Sometimes we abbreviate it like that. So when you give antibiotics, the antibiotics must narrow down to this bacteria without harming the host cells. And that's what we are calling selective toxicity. We shall talk about it when we'll be looking at antibiotics, and we shall see what some of these things mean. So how does bacteria multiply? They multiply through what we call binary fission. Binary fission is asexual. So bacteria multiply through asexual methods or sexual means. This is what we are referring to as binary fission. So binary fission is just dividing. For example, if this is one bacteria and it has maybe, let's say, 20 organelles in it, it will just synthesize other organelles. It will synthesize other organelles and divide itself. So that this one will have 20 and this will also have 20. And then at the end of the day, you'll have two bacteria now here. So you'll have bacteria A and bacteria B, of which B has come from A. So that is binary fission. just divide. The cell divides. So when we talk about binary fission, it's just dividing the cell. When it comes to classification of bacteria, there's a number of things you can look at in terms of classification. One of the things we look at is the shape. Another thing we look at is the gram stain. Gram stain. Another thing we look at is oxygen demand. Oxygen demand. So when we are using shape, we can have bacteria which are spiral in shape, which are spiral. Spiral is something like this, those kind of structure. Then we can have bacilli. Bacilli are usually rods. A rod is something like this. Then we normally have the cocci. Cocci, this one are spherical. So when we are looking at the shape, we can have spiral, we can have bacilli, which are rods, then we can have the cocci, which are spherical. We shall see that this spherical also will narrow down and see if it is just two spheres, those two spherical bacteria, they are called diplococci. So apart from being spherical, they can also exist in pairs. They can exist in clusters, so many sphericals together. Sometimes they can exist in a line, just like that. So we shall see as we proceed. So in terms of shape, we have three. You can have the spiral, what is referred to as the spiral shapes. Then we have the rods, examples of the bacilli. Can also have the spherical ones, which are referred to as the cocci. When we look, when we classify basing on the gram stain, this is a wide, broad classification which divides all bacteria into two. So we just have one, which is gram positive, and we can have two as gram negative. As we proceed, you will realize that in gram positive bacteria, the difference between these two comes when it comes to their cell wall. When you look at the structure of the cell walls of these two bacteria, you will find a difference. This one, which are the gram positives, they have a thick peptidoglycans. This is a component in the cell wall of a bacteria. So the gram positives one, they have a thick peptidoglycan, while the gram negatives one have a thin peptidoglycan in their cell walls. Therefore, these ones, by virtue of having a thick peptidoglycan, they stain this stain This stain is purple. So gram-positives having a thick peptidoglycan is able to take the stain, while these ones having a thin peptidoglycan in their cell wall, they will stain red. Remember, red is a counter stain, saffron. When you are doing the gram staining procedure, you realize that saffron is red. So the gram stain, which is purple, is washed off from the gram-negative bacteria because they have a thin peptidoglycan, thin. While the gram-positives have a thick peptidoglycan in their cell wall, which is able to pick this stain. So when it picks that stain, we call it gram-positive. And if it fails to pick that stain, we call it gram-negative. So those that take up the gram stain, which is gram-positive, they appear purple when you look through a microscope. But those that are gram-negative, they will appear red. So gram staining is a technique, it's a laboratory technique that is used to identify bacteria, whether it is a gram-positive or a gram-negative, and classify it accordingly. So another issue we can look at when classifying bacteria is the oxygen demand. Here we have two areas. You can have aerobic bacteria and anaerobic bacteria. Aerobic bacteria are those bacteria which survive in oxygen. They survive in oxygen, meaning if you remove oxygen, then their survival become hampered or interfered with. A good example here is Pseudomonas aeruginosa. Pseudomonas aeruginosa is a good example of aerobic bacteria. Anaerobic bacteria are those bacteria that can survive without oxygen. Some Clostridium species belong in this category. So those are bacteria that they don't rely on oxygen to survive. We call them anaerobic bacteria. Those that rely on oxygen to survive, we call them aerobic. A good example is Pseudomonas aeruginosa. So oxygen demand, availability is also used to classify the bacteria. When you classify bacteria based on shape, these are some of the things you will see. We said those that are spherical are known as the cocci. The cocci. Here is where you'll find bacteria like Staph aureus, streptococcus, strep pyogenes. This one is very common when it comes to infection of the throat, what we call pharyngitis. This one here. And again, it's also associated to rheumatoid fever. Rheumatoid fever. This is an issue that affects the heart. This one affects the heart. What normally happens is, this bacteria here, Once you get this infection, which is common, the commonest cause of throat infections, the pharyngitis, throat cough, it mimics the tissue, the human tissue. It has some proteins on its wall, some proteins on its wall. The wall of these strep pyogenes has got some proteins which resemble some of the proteins in the host tissues. Host tissue being the human being, particularly in the heart, sometimes in the kidneys, and so on and so forth. So when this bacteria gets into the body and it has some proteins which mimic those that are found in the human being in some parts, particularly the heart, the kidney, and others, the human body will respond by producing antibodies. So when the human body produces those antibodies, they will come to attack these proteins. And by attacking these proteins, because these proteins are similar to those that are found in the heart and the kidney, which means those ones in the heart will also not be spared. So as the antibodies come to attack these proteins, they will attack everything. Those that are produced by the bacteria, as well as those that are found in the heart and the kidneys. That's why you'll get this patient presenting with rheumatoid fever, and this one presenting with glomerulonephritis, which is autoimmune related. That type of immunity is response is what is referred to as type 2. That is type 2 hypersensitivity reaction. That was by the way. So when bacteria are of a spherical shape, they are called cocci. When they are rods, we call them mostly bacilli. Then if they are in a spiral shape, they are called spirochetes. That is an example. So Streptococcus pneumoniae causes pneumonia. These are just examples of spherical cocci. You see it exists just as two. Then we have this one that is in a linear form. Example is the streptococcal pyogenes, what we have just talked about. Staphylococcus aureus, they appear in clusters. So if you do microscopy and you find spherical shaped bacteria that are appearing in a cluster like this, that cluster, just know these are likely to be Staphylococcus aureus. And you just find two, then that is most likely streptococcal pneumoniae. You see spheres which are in the linear. Linear is just something like a line, just like this. Spheres, but they are appearing in a sort of a line. Then mostly they are Streptococcus pyogenes, and so on and so forth. When you do a microscopy and you see the rods, so An example can be Bacillus anthracis. Another example can be Salmonella enterica. Another example can be Clostridium, the Clostridium species. They are also rods. When it comes to spiral shaped, you can find Vibrio cholerae. This one causes cholera. This one here. You can also get Helicobacter pylori. This one here, which causes PUD. PUD is just peptic ulcerative disease. PUD. PUD. Just peptic ulcerative disease caused by Helicobacter pylori. Sometimes you just abbreviate it as H. pylori. A very common test in our hospitals. Then another spirochete is just Treponema pallidum. We call it T. pallidum in short sometimes. We call it just T. pallidum. Treponema pallidum causes syphilis. So those are just examples of bacteria that are spiral shaped. This is a cell wall of two sets of bacteria, a gram-negative and a gram-positive. So this is what I was talking about in the previous slide. When you look on the cell wall of a gram-negative bacteria, you will realize that it has a thin peptidoglycan. This purple thing here is what we call peptidoglycan. It forms the cell, forms the component of the cell structure. So a gram-negative bacteria has a thin peptidoglycan. And that's why it doesn't stain the gram stain. It doesn't pick this stain. But when you look at a gram-positive bacteria, it has a thick, the whole of this here. So this one is thick compared to this one, which is thin. So that is one of the differences between a gram-negative and a gram-positive bacteria. In terms of their cell walls, gram-negative has a thin peptidoglycan, while a gram-positive has got a thick peptidoglycan in their cell wall. So it is this peptidoglycan that picks the stain, the gram stain. The gram stain. And that's why it will appear purple, while this one will not pick because the peptidoglycan layer is very thin. So that is one of the differences between a gram-positive bacteria and a gram-negative bacteria. Another thing that you can see here is that a gram-negative bacteria has got two membranes. There's a membrane here and there's a membrane here. While gram-positive has got just one membrane here. So a gram-negative bacteria has two membranes, while a gram-positive bacteria has one membrane. So this is the outer membrane, while this is the cytoplasmic membrane. while this one, the gram-positive, has only cytoplasmic membrane alone. So what is the significance of those two differences? It tells you that when it comes to management of these two bacteria, then the gram-negative ones are stable. They are somehow stable, meaning they are somehow difficult to manage compared to gram-positives because of this extra layer here, which is the outer membrane. So the antibiotics find it difficult to cross, because if you want to kill this bacteria, then you are targeting some organelles, and organelles are found within the cell, inside the cell. So they are inside here. But now you have to penetrate through this outer membrane with your antibiotic to target an organelle here. For example, if you want to attack a ribosome here, if you want to stop protein synthesis of this bacteria on the ribosome, then you have a lot of work to do to penetrate that wall. Or just if you are just targeting the DNA, if you are targeting the DNA of this bacteria, your drug has to cross this layer here. And that crossing is where the issue is, because this cell wall, cell membrane poses a challenge. Therefore, on this outer membrane, there are some small channels which we call porins. So that is now where we normally target with our antibiotics. So the antibiotics will have to use these tiny openings, channels on that outer membrane for you to kill these bacteria successfully. So antibiotics must have very tiny molecules so as to be able to pass through these channels we are calling the porins. Most antibiotics that have large molecular structures cannot penetrate, unlike the gram-positive bacteria. So the gram-negative bacteria are somehow difficult to kill compared to the gram-positive bacteria. Something we need to mention also on the gram-positive bacteria is that they are very sensitive to beta-lactamase, beta-lactam antibiotics. Example of beta-lactam antibiotics is just penicillins, and also have cephalosporins. These antibiotics have a structure called beta-lactam, which is just a ring. It resembles another structure that is very essential in the formation of peptidoglycan, a layer of these bacteria. So we have something called penicillin binding proteins, that is PBP. These proteins are found on the gram-positive bacteria, and it binds on this ring when forming the peptidoglycan layer of that cell wall. So these antibiotics have a ring which is similar to the one that is used in the formation of this layer we are calling the peptidoglycan. So these proteins, which are sort of enzymes, they bind on this beta-lactam, which is artificial from these antibiotics, and incorporate this ring into this layer. And because this layer, this beta-lactam is not the original one, then the structure that is formed will not be valid. So the structure of the peptidoglycan formed on the cell wall of these antibiotics will not be functional, and therefore the bacterial organelles and other substances inside the bacteria will be exposed, and therefore that bacteria will just die. And that's why some of those drugs are known as bactericidal. Sometimes some bacteria, gram-positive bacteria, they produce an enzyme. That enzyme is called beta-lactamase. This enzyme normally digests this beta-lactam that is found in the antibiotics, therefore rendering these antibiotics weak. I repeat, we are saying that some gram-positive bacteria produce an enzyme called beta-lactamase, which digests this beta-lactam in these antibiotics. And once this ring called beta-lactam is digested, this antibiotic will be weak, and therefore it will not be functional. So that is why we have other formulations. We have other formulations of antibiotics that they add another molecule. For example, if you have amoxicillin, then you add clavulanic acid to give you amoxiclav. So the function of this will be to give you the beta-lactam ring, which is a false one, that will be binded by the protein binding that are found on the wall of that bacteria, while this one will deal with the beta-lactamase enzyme. This is an enzyme. So the beta-lactam is a ring which looks like the one found on the peptidoglycan wall of this bacteria, which these proteins bind on it because it resembles the ones found here, and incorporate them in the cell wall, making a very weak cell wall, and therefore killing the bacteria through formation of a weak cell wall. For the bacteria that produce a beta-lactamase enzyme, we have antibiotics with this added molecule which deals with this enzyme. So this clavulanic acid inactivates, blocks this enzyme. Therefore, it cannot interfere with this beta-lactam ring. Therefore, our antibiotic will be effective in that manner. So that was just, by the way, when it comes to management of gram-positives and gram-positive bacteria. This still shows the differences between the cell walls of gram-positive, which is this one, and gram-negative. What you see in yellow here is just an extra outer membrane, which is lacking in the gram-positive bacteria. So how does bacteria penetrate into tissues? When you consume a bacteria, let's say through water, it will get into your body and then it will penetrate deeper into your tissues. They exploit the cellular machinery by secreting this enzyme. So they secrete degrading enzymes. Degrading enzymes. So it is these enzymes that will degrade our extracellular matrix framework, and then it will enable just these bacteria to pass as they penetrate. So they just invade and they secrete these enzymes which degrade your tissue, and then they also evade, they escape the immune surveillance of the body. And that is the process we call invasion or invasion, depending on how we pronounce it. So that is how these bacteria penetrate into your body and they spread locally as well as systematically. And how does the bacteria cause infection? We said not all bacteria are harmful. Some are helpful in the body. So those harmful ones, when they enter your body and escape your immune defenses, they will multiply. They will multiply through binary fission. And they damage your tissues by releasing toxins. You shall see. Bacteria release toxins. There are two types of toxins. We have exotoxins, and endotoxins. So different bacteria produce different types of toxins. Exotoxins and endotoxins are produced by gram-negative bacteria, while gram-positives produce exotoxins only. So what we are saying is that gram-positive bacteria produce exotoxins only, while gram-negative bacteria produce both exotoxins and endotoxins. So what is the difference between exotoxins and endotoxins? These ones are produced in the environment, are secreted actively by the bacteria into the environment within the tissues, while endotoxins are found within the bacteria and they are only released during lysis. That is when the bacteria dies. So that's the difference. So bacteria can get into your body, as you mentioned, through various ways. Can be through breaches in the skin, when you have a wound, or when you have you have a bite, if somebody bites you, you can also ingest it through contaminated food and water, or just through inhalation in the airborne droplets. So that's how the bacteria get into your body and multiply. So we had mentioned about endotoxins and exotoxins. Endotoxins, they are mostly produced by gram-negative bacteria, while exotoxins are produced mostly by gram-positive bacteria. Remember, gram-negative bacteria produce both endotoxin and exotoxins. But the gram-positive bacteria produce exotoxins only. So this is just a summary table of some key toxins that are produced by some bacteria. We just picked an example. For example, Clostridium botulinum produces what we call botulinum neurotoxins, which causes flaccid paralysis. While Clostridium tetani produces tetanospasmin toxins, which causes spastic paralysis, what you call tetanus. Then Vibrio cholerae produces cholera toxins, and that causes cholera infection, that is diarrhea. Staphylococcus aureus produces what we call toxic shock skin toxins, that is 1 and alpha, which causes toxic shock syndrome, that is the Staphylococcus aureus. The diphtheria produces the diphtheria toxins, and that is related to throat infection, while the gram-negatives produces this type of toxins that causes sepsis. So that is just a continuation of what we have already mentioned in terms of those toxins. Cytotoxins kill cells through causing necrosis and tissue destruction. And the exotoxins cause this type of exotoxins, which we are calling the neurotoxins. They block neurotransmitters, while the enterotoxins, they alter ion transport in the GIT, and that causes diarrhea, and so on and so forth. So we had talked about the concept of molecular mimicry, whereby certain bacteria, for example, the streptococcal pyogenes, which produce antigens that resemble human tissues. Some of these antigens are known as M proteins. So they are produced by streptococcal pyogenes, what we refer to as streptococcus. And these proteins, they share structure